let's talk about vacuuming my house. I promise this will relate to genetic testing in a moment. So when my 10-year-old vacuums the living room, that means something different compared to when I vacuum the living room. He vacuums about 60% of the exposed carpet, running over the carpet only once. Whereas my perfectionism leads me to vacuum over 99% of the exposed carpet, carefully overlapping each vacuum track multiple times to make sure I'm picking up as much as possible. We can both say the living room has been vacuumed, but the thoroughness of what is picked up from the carpet differs significantly between us. The same is true for DNA sequencing technologies and how they are used by different labs to sequence genes. The regions of each gene covered and not covered, and the thoroughness of coverage over each region, known as sequencing depth, differs between labs even when the labs are CLIA and CAP accredited. If the sequencing depth is too shallow over a region, detection of variants is unreliable or not possible. So differences in gene coverage and sequencing depth can be the reason why one lab's test is better at picking up genetic variants than another. But it is not yet a regulatory requirement that labs make these types of test details transparent. Sensitivity and specificity measurements define something other than coverage. As of April 2021, when this video was filmed, most genetic testing labs describe the regions of each gene they cover with generalized, non-specific summaries. Is it time we raise our expectations for more technical details from labs? After all, these types of detailed metrics are important to understanding the likelihood of false negatives from a laboratory's test for our patients. With the support of the Utah Cancer Genomics program of the Utah Department of Health and funded by the CDC, we are asking laboratories for more detailed descriptions of their tests, which will be collated at testwisely.org. Other technical details about coverage and depth are helpful, but let's at least start with nailing down the exact maps of confident next-generation sequencing coverage in genes from different labs. If we go back to the vacuuming example, we want to know what percentage of each room is being vacuumed, where it was vacuumed, and how thoroughly it was vacuumed.